Hey everyone, we're back with Victor. He just finished giving a wonderful talk earlier today about Huntington disease and how it's been affecting his family and how he's working to learn more about the disease and how to cure it, and other poly-Q diseases like SCA1, which he's been working with mostly in his beginning of his PhD. And so we just have a couple more questions for you that we'd love if you could answer. And so the first question that we have is, you mentioned a little bit about clumping. And does the amount of those clumps affect the impact of the disease?
Yes. So clumping, it's very controversial in the field. Some people think the clumps are good. Some think the clumps are bad. You know, in the olden times, we thought clumps, big clumps, cells die. Other people think the clumps are putting all the garbage, all the proteins in one area, so they don't affect other areas of the cell. So there's debate about the clumps. I use the clumps as an example because it's the easiest way to understand the disease, about how these clumps kill, or we think kill. But there are some scientists that are for and against clumps. So big clumps are bad, big clumps are good. The field is constantly changing with science when we get new discoveries. I personally think clumps are bad, but you know, we still need to know more about the disease and what it does. It's hard to know, you know, if you sequester all the protein in one area, you know, what's the normal protein? Because some of it folds correctly, but we don't know what that stuff's doing. So to answer your question, we don't know.
Okay, so following up then, going a little bit different direction. So that clumping action was because you have a bunch of cues in a row and the cues are dragging them together, we think. And those cues come from the CAG repeats that you talked about at the beginning. And you let us know that if you have too many CAG repeats, you can see this disease income. But what if you have too few? You put the healthy range at about 5 or 6. What if you have fewer than that number of CAG repeats?
Yeah, so for example, I could use mice. Mice have 2 normally. They have 2 of them. So they still have some. They don't have 0. And in any similar protein in this and any other organism that has it, they still have these cues. So there's no organism that doesn't have any cues. So the cues play some role. What role that is, we don't know, but they do play a role somewhere. So some scientists have tried getting rid of the cues using CRISPR, and the results vary depending on the lab. So it's still up in the air. If we just cut the cues out and not have disease, some say you survive, some say you die before you're born. I don't know how you get such big results from doing the same thing, but they do. So too few can be a problem, and too many can definitely be a problem. So you gotta be in that healthy range. And the cues actually fluctuate between parents. When your father passes the disease on to the child, the cues can actually get longer. But when the mother passes it on, they can get smaller. So my grandfather died after my aunt because he passed on his disease, but it went bigger for her. So she had it earlier than him. So the cues they grow, they shrink. It's very, yeah. And then also if you're at 37 cues, 1 cue above that 36 for Huntington's, you are in between. Like you have the disease, so it's dominant, you have it, but you might not show it still. So it's like the 36 to 39 range of Huntington's. It's like an intermediate disease, smaller effect. You still have it, but not as deep as the other repeats. So there's work on that. The more repeats you have, the more severe the disease is.
Interesting. And so on that same, still thinking about, we're talking about the amount of cues. If we think about like the amount of diseased cells, does the amount of diseased cells also impact the severity of the disease in the patients?
Okay, so the diseased cells, these are the neurons that are affected. So for each poly-Q disease, a different brain region is affected. For Huntington's, it's the basal ganglia region, that region in the middle of the brain that had a hole in it in the picture. In SCA1, it's the cerebellum, which is in the back of your brain here. And so, each cell, they affect certain cell types. In Huntington's, it's the medium spiny striatal neurons. And in SCA1, it is the Purkinje cells. So over time, those cells will start dying and getting the disease. And so only those cells, though, are usually affected. So those are the disease cells, but there's other pathways that those cells talk to and they connect to things, and those get affected. They don't die, but they're losing connections, or they're being interrupted. If you remember, every cell expresses these proteins for Huntington's and for SCA1, but not every cell is dying. So are they diseased? Are they not? How diseased are they? In SCA1, the cerebellum's affected, the back of the brain, but if you look at the cortex, these other areas of the brain, there's slight decay. There's slight effects there because it's expressed there as well. Why then only those cells there are dying? We're still working on that. We're still trying to figure out why certain areas, when it's expressed all throughout your body, why certain regions are more prone or more sensitive to it. Are they just more sensitive, or are they unprotected? Is there a protein expressed everywhere else in the body, but not in that region? And so everything's protected but that region? We're trying to discover that. And of course, to answer, go back to your question on the number, sure, the more cells that have and died, the more severe your disease will be. But it shouldn't be thinking of the number, it should think of the region and types of cells that are affected, really, and how we can stop that from happening there. If we stop the cells in the cerebellum from dying, that's great, but then maybe you live longer and now the cells in the cortex or other regions now start dying. Maybe it took longer for them to die. Maybe they were diseased, but at a lower rate, slower rate, and they will die just slower.
So following off of that, for SCA1 or Huntington's or any other polyQ, do you still see that protein clumping even in the cells that you normally don't think of as being affected?
No, the protein clumping really is in those cells that are dying. And that's what led to people to believe that protein clumping led to cells dying. But you have all these other connectivity issues and other things happening before the cells die. Like I showed that timeline, you have at 5 weeks, they can't walk correctly, and then they have, their thought process is affected earlier than the cells dying. So it's not that the clumps are killing the cells and that's the cause of the disease. Maybe the clumps are affecting different biological pathways that then lead to the cell dying, stuff like that. We're still trying to figure out, maybe they're clumping all the stuff together and some of it is escaping and being toxic, when you're trying to clump it all together to prevent the toxic function of it. Or maybe that clump kills it. Still trying to debate that, still trying to figure it out exactly. For SCA1, we know what it does normally, so we can more easily assume what's happening. But for Huntington's, we don't know what it does normally. So we don't know, we still can't, we can make a hypothesis, but we still don't know. It's very, very annoying. Trying to figure out what this protein does. It's weird to say that we don't know what a protein does normally, but we know when it's bad, we know what happens. You would just go, oh, it does the opposite, but no, it doesn't just do the opposite.
So you talked a lot about the impact of these cells dying, but when I think about a lot of cells in the body, like they can regenerate. Is that not the case for these diseases?
Correct. So neurons are very special in that once they've been made, they don't divide anymore. Your brain doesn't keep growing. Your brain is a certain size. And once you reach that maturity, it's done. The cells stop dividing. So if you lose a neuron, it's gone. So that's why you have to really take care of your brain. You can't damage it because that damage is permanent. You can't regrow it. So if you think of a simple A to B to C, one neuron, another, and another in a linear pathway, if B is gone, A can't go to C anymore in that straight pathway. So maybe it has to go around some other way, like we talked in memory a few weeks ago, how you get these memories by these neurons. If they're controlling your body and one neuron tells your arm to move and it's gone, well, your arm's not going to move anymore when your brain tells it to. It's going to move on its own kind of stuff. So sadly, these neurons cannot regenerate on their own. And that's why step 2 is so hard. How do you regenerate neurons? How to regenerate pathways? You have to take a lot of different drugs and other things to force those cells into a state that they can divide again. And getting that to happen is incredibly hard. We do it in the lab in cell culture, and so we force it to. And it's easier because it's on a little petri dish and we can do it in lab, and we just throw a lot of stuff at it to force it. but it's a lot harder in a living organism to make that work. And then if, how do you target just certain cells, certain very small cells in a certain small region of the brain to regrow and not every other brain cell? Because your skull doesn't grow anymore. If you get your whole brain to double, that's going to be bad. So how do you target very small cells? Very challenging.
So this is a question that wasn't from the students, but I'm going to try to tee you up on it anyways. So Dan, at the end of his talk today, also brought up how the question that he's looking at is something that requires the efforts of scientists across a range of fields. Would you feel that that is the same for your study of SCOT1 now or Huntington in the future?
Entirely. There are many, many labs out there doing many great things in SCOT1, in Huntington's. I'm currently qualifying for my PhD, and all these papers I'm reading are from many different labs all across America. And you read these things that they do, and so these other people out there influence what you do here. And then I'll maybe just come up with a discovery that I put out there, and other people will read it, or they'll talk to me, and we'll discuss together. My lab here at Yale is currently collaborating with 3 or 4 other labs right now on different projects. So within our university, we're collaborating, and then our labs also collaborating with other labs and other universities in like Columbia, which is in New York, I believe. I think my Ivy League college is bad. I think it's in New York, and other like in Minnesota, and we have a collaboration in like San Diego. So we're collaborating very much because, like I said, you can't cure this on your own. You can't do it on your own. In 27 years since the discovery of the disease, we still haven't cured it. And that's with many people working on it. And so that's why I hope that one day these students here, maybe some of them will join and, work with me on it. And it's going to take a long time. I expect in 20 years, I'll still be working on it. And if by luck I cure one disease, I got 8 other ones to work on. And if I cure those 8, well, there's Alzheimer's, there's Parkinson's, there's many diseases out there. My work's never going to be done. There's always going to be something to discover.
Well, this has been fantastic, Victor. It was a great talk, and I think the students will appreciate having some more answers to their questions. And really, thanks so much for giving us this time today.
No, thank you for having me. I'm really excited to do this. It was a great honor for me to talk at your guys' event.
All right, so we're back with Dan, who gave us a really excellent talk today, talking about symmetry in nature and molecules and what happens when it's not there. So we're going to follow up with some of the questions that y'all asked, just to make sure we get a chance to get to as many of them as possible. So Dan, first, you mentioned about the left-handedness and right-handedness of DNA, but is it possible that, what if like artificial DNA was created to go in the opposite direction? Could that happen, and what would that mean?
Yeah, so that's a really good question. Thing is, nobody has found this DNA that faces the other way in any real organisms, but it has been created in the lab for the exact purpose of studying handedness in molecules. But no, there isn't any DNA like this in any living thing. So if the DNA was really the other way, it could work in very much the same way, as long as all of the other molecules that DNA interacts with, including proteins, also had flipped-handedness. So if everything is exactly flipped, it all works. But if you just put, left-handed DNA into a right-handed environment, it's not going to work at all.
That's actually what I was thinking about as a follow-up question, because we talk about these molecular scissors a lot, right? And we know that the left-handed, right-handed scissors are sometimes difficult to use. I mean, that's kind of a silly analogy, but yeah.
Right,
so
no, the
entire system.
Mm-hmm.
Interesting. Yeah. And so on the topic of this left-handed versus right-handed and the entire system needing to be different, whenever you showed that picture about the sort of ocean with like left-handed, right-handed stuff, weren't the left-handed molecules also making more of themselves? And how did that happen? Yeah,
so that's one of the biggest problems with, the idea that there was a right-handed molecule that just made more of itself, because you ask, well, okay, but what about the left-handed molecule that made more of itself? So actually, there is no good answer. Nobody knows. That's one of the greatest mysteries in all of science. It's possible that if there was slightly more of, for example, the right-handed DNA than the left-handed, then over time, because it kept copying itself, it could have overpowered the left. so much that there's basically no left-handed DNA left anymore. But how that happened and whether that happened is a complete mystery.
Do you think it has to happen with like interactions with other molecules as well? Like sort of by chance, other molecules that interact with it also were right-handed, and then that sort of just influenced those to be propagated?
Yeah, so that's possible. It's possible that there was a small imbalance in, slightly more right-handed than left-handed DNA, but also the molecules that are needed to, help copy the DNA and make more DNA also had the same sort of unevenness. And, it started from a small amount, but if you imagine that every time DNA copies itself, it multiplies itself by 2 times 2 times 2 times 2, and every time you get exponentially more DNA. So even a small difference in right-handed versus left-handed populations on the early Earth could very quickly turn into like 99.999999% right-handed.
Right. That makes sense. And so we are talking about DNA from like an early stage, and then we're sort of jumping to humans because that's what's most relevant to us a lot of times. But does this hold true for other organisms in addition to humans and animals about the handedness of DNA?
Yeah, so it actually does. The DNA is right-handed in every animal, but also in every plant, in every fungus, in every bacterium. Anything that has been discovered, any species of organism that has been discovered ever, has right-handed DNA and right-handed DNA only, which is a really big mystery. But it also, really shows how really all life on Earth is interrelated. And also it supports the idea that all life on Earth came from one common ancestor that had right-handed DNA.
I see.
Yeah.
Awesome. And are there any inherent differences between sort of the right-handed and left-handed, or are they, as far as people understand, basically the same? They're just flips.
Yeah. So the difference is only in the right-handed or left-handed DNA's interaction with other right-handed or left-handed molecules. So if you take everything right-handed and just flip it to being left-handed, it would be exactly the same. But if you only flip the DNA but didn't flip the protein, for example, then it would be like trying to shake your own hand, and it would be awkward, and not all of biology, it would break down. So it's important for things to match, but once they match, it doesn't matter if it's right or left.
I see. Awesome. Cool. This stuff is really fascinating, and I struggle with some of these. A lot of the pictures that you showed about whether or not they actually line up on each other, I have a hard time thinking about that, but it's really awesome to see people studying this sort of stuff and trying to understand what it all means and make it all make sense.
Yeah, the 3D part can be a little confusing, especially on a PowerPoint. It's easier to show this to yourself, this handedness using actual models, but unfortunately that is impossible with Zoom. So.
Yeah, well, it seems like the hand example is really good, though. I think that's really visceral for people.
Yeah, that's probably the best example is actually on our own bodies. Yeah.
Great. Awesome. Well, thanks a lot, Dan. I really appreciate it, and we'll check you next time.
All right, yeah, no problem.